Remember how we learned in earlier chapters that the enzymes that propel the biochemical reactions in our body work only in a very narrow range of temperature and pH? Well, in this segment, we'll discuss how animals maintain that narrow range of temperature within their bodies that's necessary to sustain life. We term this ability to maintain a constant internal temperature homeostasis. Although Arctic temperatures can drop far below freezing, polar bears maintain an internal body temperature of 98.6 degrees Fahrenheit, the same as humans. Normal body temperature is approximately 98.6 degrees Fahrenheit. Hyperthermia occurs when too much heat is produced or when high environmental temperature and humidity overwhelm the body's ability to dissipate heat. Disastrous consequences can result from extreme hyperthermia, called heat stroke, and death might even occur. This illustrates the danger when organisms fail to maintain an internal environment within a safe range. Although the environmental temperature range you experience every day might involve a 20, 30, or even 40-degree difference between high and low, humans can't tolerate such large internal temperature swings. Even deviations of less than 10 degrees can lead to a cascade of biochemical problems and serious health implications. For mammals, the risk of getting too hot include reduced enzyme activity and, at extreme temperatures, the complete breakdown of enzymes. Excessive water loss, too, can occur, leading to numerous problems, including organ failure. For these reasons, heat stroke can be life-threatening. At the other extreme, hypothermia, or excessively low body temperature, can cause difficulties in coordination and movement along with disorientation and confusion, irregular heartbeat, and ultimately death. Similarly, disastrous consequences can result from the failure to maintain a constant internal environment in other ways, including blood sugar levels, blood pH, and tissue concentrations of oxygen and carbon dioxide. A fundamental characteristic of organism function, homeostasis, is the body's use of physical and chemical processes to maintain a constant internal environment, even in the face of changing external and internal environmental forces. With multicellularity and increasing size, an animal's internal environment takes on greater importance than the external environment in influencing cell functioning. In vertebrates, this internal environment consists of the extracellular fluid, also called the interstitial fluid, that fills the space between cells bathing the cells. Variables such as temperature, water solute balances, pH, blood sugar levels, and oxygen and carbon dioxide concentration in blood and other tissues are maintained within narrow ranges by the activities of cells within the organism's tissues, organs, and organ systems. So just how does homeostasis work? Well, many creatures have differing ways to maintain body temperature. For example, by adjusting its postures, a red-veined drop-winged dragonfly is able to cool itself on a hot day. For animals to maintain homeostasis with regard to a particular physiological variable, that variable must have a set point, a target value or range to which the organism can return. Most vertebrates have set points for a variety of physiological variables, including body temperature, water solute balances, blood sugar levels, blood pH, and tissue oxygen and carbon dioxide concentrations. In the face of changes in its external environment, how does an organism maintain its homeostasis, its constant internal environment? Well, the most common method involves the regulatory mechanism of negative feedback, in which sensors detect a change in the internal environment and trigger structures called effectors to oppose or reduce the change. This cycle of detection, response, and change is called a negative feedback loop. Think of the way the heating system in your house or apartment is controlled by the thermostat and the set point that you give the thermostat. This is a mechanical example of a negative feedback loop. It's important to note that while some groups of organisms called regulators maintain homeostasis for a certain variable, just as we've described for temperature, other organisms called conformers may have no set point for that variable, and the variable may fluctuate with external changes. Animals differ in the traits in which they are regulators and for those which they are conformers. Most fishes and shellfish, like shrimp for example, are conformers with regard to the temperature of the surrounding water, but are regulators when it comes to the concentration of salt in their blood and tissues. In addition to negative feedback systems, organisms also have a small number of positive feedback systems, in which deviations from conditions normally found in the external environment cause an increase or acceleration of the change in the same direction. Such systems push the body away from homeostasis and often initiate cascading processes that increase the rate at which the system deviates from the normal range. For this reason, positive feedback systems, if unchecked, can become highly unstable. An example of a positive feedback system is the blood clotting process. Injury to a blood vessel causes platelets, which are cellular fragments involved in clotting, to release their superglue-like contents, which begin to seal any rip or tear in the blood vessel. Within the bloodstream, the contents released from a platelet have another effect as well. They cause other platelets to release their contents. This is a positive feedback system because the release of some blood clotting molecules into the bloodstream can cause the release of more such molecules, which causes the release of still more. The result is that a dangerous situation, that is, injury to a blood vessel, is quickly remedied. For many animal species, the control of temperature, called thermoregulation, is an important component of homeostasis. Some of the important benefits to controlling body temperature include enhancing enzyme stability and efficiency, for, as we've just seen, a small change in temperature often significantly reduces the rate at which an enzyme catalyzes a reaction. This type of regulation gives organisms more independence in the timing of their daily activities and, more importantly, resisting freezing. In thermoregulation, the biggest distinction among animal species is how they generate their body heat. Endotherms, sometimes described as warm-blooded, generate their heat internally within their own bodies. Most mammals and birds are endotherms. Ectotherms, sometimes described as cold-blooded, get their heat primarily from the environment, which is usually the sun. Invertebrates, fishes, amphibians, and reptiles are all ectotherms. There are costs and benefits associated with being an endotherm versus an ectotherm. Between 27 and 36 degrees centigrade, for example, endotherms are able to maintain a constant internal body temperature without any energy expenditures. Within this range, called the thermoneutral zone, their resting metabolic rate can remain constant as they maintain their constant body temperature by using passive methods of regulation such as heat loss through the skin. In ectotherms, on the other hand, metabolic rate increases continuously across this range of temperatures. Below the endotherm's thermoneutral zone, the situation is reversed. Metabolic rate increases significantly as they must use active heating to maintain their body temperature. At these same colder temperatures, metabolic rates in ectotherms decrease as they let their body temperature drop along with the ambient temperature. Endotherms and ectotherms, as we mentioned, are often distinguished as warm-blooded and cold-blooded, but on closer investigation, these terms don't always accurately describe the two groups. For example, some organisms, such as humans, are homeotherms, meaning that they maintain a relatively constant body temperature. Other organisms, such as lizards and hibernating mammals, are heterotherms, and their body temperature at times fluctuates as the environmental temperature changes. As a consequence, animals are most accurately categorized by describing their source of heat, internal or external, and the degree to which they maintain a constant temperature or have a temperature that fluctuates. Whether or not the animal generates its own heat and maintains a constant body temperature, all animals exchange heat with the environment. Ultimately, an organism's temperature is the function of heat produced by its body in conjunction with the heat transferred from the environment to its body or from its body to the environment. This heat exchange can occur in four ways. First, conduction. Second, convection. Third, radiation. Or fourth, evaporation. Numerous physical features have evolved in organisms that influence their body temperature. These include body size, surface area, and levels of insulation. An organism can also use behavioral strategies to regulate its body temperature. Animals also employ many physiological methods of thermoregulation. One cellular method of temperature regulation is particularly important to human babies. It takes place in a special type of connective tissue called brown fat. When brown fat cells oxidize their fat, they don't generate ATP. Instead, a special protein causes protons to leak directly across the mitochondrial membrane, rather than passing through the enzyme that synthesizes ATP, causing the production of heat rather than ATP from the fat breakdown. Organisms, whether they live in the water or on land, require water, but they can't take in too much or they'll burst, and they can't lose too much or they'll shrivel up and die. We've discussed osmosis in previous chapters, and osmoregulation is an important component of homeostasis in animals, and it's the regulation of water content and of the concentrations of dissolved solutes that influence osmosis. The most abundant solutes in animal fats are sodium, chloride, potassium, magnesium, and calcium ions. Although a variety of mechanisms of water regulation have evolved in animals, there are just four ways by which animals get water: drinking it, eating foods that contain it, absorbing it through osmosis, and as a byproduct of cellular respiration. Similarly, there are four ways that animals lose water: urination, defecation, evaporation, which includes panning, breathing, and sweating, and osmosis. Different strategies have evolved for coping with diverse osmoregulatory challenges. Most invertebrates that live in salt water are osmoconformers, meaning that they let the solute concentration of their body fluids reflect that of their environment. Most vertebrates, on the other hand, are osmoregulators, maintaining their fluids and solute concentrations within narrow ranges that differ from those of their environment. The kidney is an organ in invertebrates that helps maintain homeostasis by regulating water balance and solute concentration in body fluids. It accomplishes this by filtering blood. As much as 2,000 liters, or 275 times the total volume of blood in your body, pass through the human kidneys every day, and resorbing water and other substances needed by the body. The kidneys accomplish three primary functions. The first is filtration. As blood passes through the kidneys, it's filtered. During this process, water, ions, and other dissolved substances are removed from the blood as a filtrate, leaving behind just cells and large proteins. The second is reabsorption. The filtrate then is processed and modified as water, nutrients, including glucose and amino acids, and some ions are reabsorbed into the blood, concentrating the filtrate. And third, secretion. Following the concentration of the filtrate, the substances not needed by the body are excreted in the urine. So to summarize, human kidneys filter blood, reabsorb water and solutes, and excrete waste. A human has two kidneys, each about the size of a fist, located on either side of the spine just above the waist. Each kidney is made up of approximately 1,000,000 nephrons, the structural units that accomplish the work of the kidneys. A nephron consists of two basic components: a nephron tubule and a mass of blood vessels that work together to accomplish the task of filtration, reabsorption, and secretion. The blood filtering unit of the nephron is a mass of capillaries called the glomerulus, and the ball-like structure that surrounds it is called Bowman's capsule. Each Bowman's capsule is connected to a single long urine-producing tube that excretes its filtered fluid into a collecting duct. So the nephron is the fundamental unit of the human kidney. 1 million nephrons enable the human kidney to regulate water balance and solute concentration in body fluids while producing urine. The human kidneys are so effective at filtering blood and concentrating the many waste products of the metabolism in urine that it's possible to detect the use of many drugs through urinalysis. Most drug screening urinalysis doesn't actually involve testing for the presence of the drugs themselves, which may remain in the body for only a short time. Instead, the tests look for the presence of chemicals called metabolites that result from the breakdown of the drugs. Metabolites have been identified for many drugs, including ecstasy, cocaine, methamphetamines, and marijuana.